After 35 years of research, scientists have only just begun to understand the importance of the human endocannabinoid system, or HEcS. The discovery of the HEcS is arguably the most important discovery in human physiology in the late 20th century. Research has revealed that the HEcS is responsible for maintaining and controlling the body's homeostasis, or balanced regulation of every system in the body. It does that through two known receptors called CB1 and CB2. While the body produces its own endogenous cannabinoids, many scientists suggest that most people are now suffering from cannabinoid deficiency. Without sufficient cannabinoids in our diet, the HEcS operates at less than peak efficiency, resulting in a general decline in overall health. Supplementing our diets with full-spectrum phytocannabinoids may be an essential component to achieving optimal health. The endocannabinoid system is found in every animal except for insects and regulates a broad range of biological functions. The HEcS is a biochemical control system of neuromodulatory lipids, which are molecules that include fats, waxes, sterols, and fat-soluble vitamins such as vitamins A, D, E, and K, and others. In general, a given receptor will accept only particular classes of compounds and will be unaffected by other compounds, just as a specific key is needed to open a lock. Specialized receptors are located throughout the human body, including but not limited to in the hippocampus, which is involved in memory and learning, the cerebral cortex, responsible for decision-making and emotional behavior, the cerebellum, which controls motor functions and coordination, the putamen, associated with movement and learning, the hypothalamus, regulating appetite and body temperature, and the amygdala, which is key to our emotions. When a specific cannabinoid or combination of cannabinoids binds to a specialized receptor, an event or a series of events is triggered in the cell. This results in a change in the cell's activity, its gene regulation, and all the signals that it sends to neighboring cells. This process is called signal transduction. First detected in the brain, science now shows that CB1R are also located in many other organs, connective tissues, gonads, and glands. CB1R are not found in the medulla oblongata, the part of the brain stem responsible for respiratory and cardiovascular functions. CB1R play an important role in the coordination of movements, spatial orientation, sensory perceptions such as taste, touch, smell, and hearing, as well as cognitive performance and motivation. The most important function of the CB1R is the reduction of excessive or inadequate signaling by the neurotransmitters or messengers in the brain. By the activation of the CB1R, the hyperactivity or hypoactivity of the messengers, like serotonin and dopamine, is regulated back into balance. For example, when THC binds to CB1R, activity in the pain circuits is inhibited, thus resulting in reduced pain. Many other symptoms, such as nausea, muscle spasticity, and seizures, can be alleviated or diminished with cannabinoid therapy. CB2R are primarily associated with the immune system and found outside of the brain in such places as the gut, spleen, liver, heart, kidneys, bones, blood vessels, lymph cells, endocrine glands, and reproductive organs. For example, CBD is key to CB2R, and good evidence shows CBD is a beneficial therapeutic strategy to lessen the impact of inflammatory and neuroinflammatory diseases. However, studies now show that it also plays an important role in the signal processing of the brain. A third receptor that gets little attention is the transient receptor potential vanilloid type 1, or TRPV1. The function of TRPV1 is to detect and regulate body temperature. In addition, TRPV1 is responsible for the sensations of extreme external heat and pain and is subject to desensitization. Therefore, if continuously stimulated, the pathway will eventually slow down or even stop. This raises therapeutic possibilities for agents to effectively treat certain kinds of neuropathic pain.